Professor Mike Dixon is in a place where cannabis is legal from an adult use standpoint, and from a market standpoint, Mike is in that unique environment, but also unique in terms of an environment is the expertise Mike has as a professor for a long time on plants, but bringing that experience to cannabis in this respect from the University of Guelph and the Cannabis Center of Excellence. Mike, thank you for joining us, Mike.
My pleasure, Keith. As always, it's good to hear you and good to see you. And I come to the cannabis story from a completely different sector of science. Horticulture, to be fair, plant science, plant physiology, plant environment interaction. But my field of research is space exploration and specifically life support, biological life support, food, which determines how far from Earth we can go and how long we can stay, and we have to take green plants with us. And so we've been doing for the last 25 years or so at Guelph, at the University of Guelph, building a program of developing the technologies required to reliably and indefinitely produce plants for food in some pretty harsh environments. There's some obvious technical spin-offs here on Earth in the agri-food sector, especially in northern Canada, where we have a food security issue, and in other harsh environments around the world, like the deserts of the Middle East, etc. So let me see, I'm getting a... So if you want to start switching slides for me, John, here's part of my playground at Guelph. We use high-intensity customized LED systems to in these small round chambers, originally hypobaric chambers, so that we could look at the influence of pressure on plants, determine how low you can take the pressure and therefore reduce the mass cost of your greenhouse on the moon and Mars, for example. Next slide. This is the latest generation of controlled environment chamber, and basically all configurations of sealed environment chambers that we grow plants in our program. They vary in size, but they have extremely high fidelity environment control in light quality and quantity, CO2 concentration, temperature, humidity, nutrients, and water. And so basically we're farming, but at a very high fidelity environment control level, and then we can play a tune with those environment variables and simultaneously, second by second, monitor photosynthetic gas exchange, so we know how the plant is consuming CO2, releasing oxygen, releasing water vapor as a source of fresh water. And of course, they make us feel good. So plants have that hort therapy element to them that is actually not to be discounted. The astronaut, I know many astronauts, and they fight over the plant biology experiments on the space station and elsewhere. So it's something that's sort of intangible, but not to be discounted. Next slide, please. So this is just a list of what we control. And as I say, that critical explanation there, that the very high fidelity control, which yields the systematic or the standardized response. So what we're looking for here is standardizing what these plants end up, their profile of medicinal compounds. The work we've been doing in the space program for the last 25 years has sought to standardize the profile of nutritional compounds so that you can predictably predict your life support requirements. So many different kinds of recipe elements for a menu selection, for example. And if you can predictably have the same nutritional composition in all the plants that you have in your recipe for the menu, then you can devise a life support system that's sustainable. That is exactly what the phytopharmaceutical industry sector requires to change the perception of cannabis to one of a conventional pharmaceutical commodity. It doesn't have that today because it isn't the same, and I suspect that a lot of the variability. In fact, I know that a lot of the variability reported by researchers and some of the work that has been presented here today is a result of the variability of the plant, the variability of the profiles of medicinal compounds, the cannabinoids, terpenes, flavonoids. There's over 500 compounds that we know of so far, and the recipe of those compounds yields, as Mechoulam says, Rafi Mechoulam says, the entourage effect of that compost of chemistry yields the medical efficacy. And we're just starting, so there's an awful long way to go in this particular science and field. But I'm sort of stealing from the space exploration technology development that yields this very high quality and predictable concentrations of nutritional compounds and applying that to medicinal compounds. Next slide, John, please. So this is just a sample of some of the kinds of plants that we look at, the food crops. And next slide so here's just a profile of some of these food crops in controlled environments. So tomatoes, lettuce, peppers, so conventional salad crops. And the difference between those two little families of curves there is that the bottom one has roughly ambient carbon dioxide levels, 400 parts per million, and the upper family of curves has 3 times that, so 1,200 parts per million of carbon dioxide. And then this is called a typical light curve of photosynthesis on the left and increasing light intensity on the bottom, and the light is all the same quality. So this is a sort of a a standardized recipe of spectral quality. But the next, just push the button again, John, and you'll see that if you just go to the 400 micromoles, which is about a fifth of full sunlight, full sunlight being roughly 2,000. So we are at a relatively low light level, but if you, without adding any more energy to the system whatsoever, just tripling the concentration of carbon dioxide, from 400 to 1,200 parts per million, it's still just a little squirt of CO2. Nevertheless, you virtually double the productivity, the photosynthetic productivity of the plant, its vegetative production, whatever. So without adding any energy, and these are the kinds of environment control, plant environment interaction issues that the cannabis industry needs to wrestle with and standardize to achieve the levels of production and standardize the production levels as well as the quality of the plant. Next slide. So here's a really simple little experiment with lettuce. Lettuce is, I submit, is not food, but we use it as a guinea pig in a lot of our research experiments because the darn stuff grows so fast. In 30 days you can have a full-grown lettuce crop ready for harvest, and so that's enough. That allows us to turn experiments around in a very short order. And this is literally a very simple experiment in a high-density modular food production system that could grow a wide variety of different crops depending on their stature. And so we're using red, white, and blue light for no reason other than, quite honestly, to have a neat picture at this point. Next slide. So here's what you get. These plants are genetically the same. I won't say they're identical. They're probably an F6 or F7 variety. So they're bred to be the same. And this is a typical designer lettuce. It's called New Red Fire. and it has a little bit of anthracene and then the leaves. But look at the remarkable difference from the white and the red and the blue. The red is virtually double the biomass of each of the other two. And there are some obvious differences in color and size. And then, of course, I have a large group of grad students in the facility. And so a bunch of the grad students and technicians got together and did a blind taste test with these plants. And with 100% reliability, they could just close their eyes and say, oh, that's the blue one, that's the red one, etc. So we've modified the size and the shape, the color, and the taste. Color and taste, by the way, are products of secondary metabolic activity in the plant, secondary metabolites, anthracene and whatever taste compounds lettuce happen to have. So in principle, this is precisely what is required in cannabis to manipulate and standardize the profile of medical compounds, just as we're messing with the taste and color compounds in this plant. We can apply exactly the same controlled environment technology to yield variations in the expression of cannabinoids, terpenes, flavonoids, the compounds of interest in cannabis. And so these are all genetically the same. And I submit then that if you have, and if we go back to the previous speaker before Dr. Carrillo, you'll notice that, sorry, not the previous slide, thanks, John. The genetics they lay the table for the plant. The DNA profile of a plant lays the table for its adaptability to environment conditions. It has the power to acclimate to virtually an order of magnitude range of environment variables for everything except temperature, really, and that's almost half an order of magnitude there. So the issue here is the standardization requirements can be yielded by high fidelity environment control. And we can steal that technology today from the space program and do a bit of an end run on all the pharmacology and biochemistry that must happen over the next couple of generations, I guess, to get more reliable standardization that doesn't require extracting these compounds or synthesizing them in some other way. I think the plant can do it for us. Next slide, John. So here we come to our familiar friend. And I still do not have a license to grow this stuff on the campus at the University of Guelph. That is pending, and I expect to hear more about that literally in the coming weeks, which will then allow us to proceed with infrastructure developments, new research infrastructure to grow plants and play with cannabis legally on the campus. It's legal to have it and grow 4 or 5 plants of your own, but it's still illegal for academic institutions to, or I shouldn't say illegal. It is very strictly regulated on what you can do with cannabis when you grow it, if you don't have a license to do research or produce it. So we're getting there. That will happen, COVID notwithstanding. That will happen in the coming weeks to months, certainly this year. So next slide, please. In the meantime, my graduate students have been doing research on the sites of our industry partners. And in this case, an industry partner that's about 3 hours away, which makes it a bit of a logistical challenge for us, but nevertheless, an ideal facility, very well run. And we've been looking at this particular technology here is a sensor that measures the water stress in the plant and can feed back to develop algorithms for irrigation management that standardize the irrigation management. Next slide. And here we're looking at lighting, introducing lighting inside the canopy of the plant. The vast majority of our environment engineering, if you will, has been trying to homogenize the environment. So homogenize the concentration of carbon dioxide, the temperature, the humidity, or vapor pressure deficit really is what plants sense, the nutrients and the water. So that's 5 of the 6 big variables. Light, attempting to homogenize. Imagine if every leaf saw the same CO2, light, quality and quantity, temperature, humidity, nutrients, and water, then every leaf would perform the same. That's practically impossible, but what we're doing here is pursuing the objective of distributing light more efficiently, photosynthetically active light more efficiently into the inner canopy. And the next slide shows some of the results of that. These data don't take the numbers to heart, other than what we've demonstrated here is that just by adding a little bit of light in the lower canopy of the plant, this is preliminary work, we haven't gone to the extent yet of homogenizing the light spectrum. Actually, that's not true. That is happening now, but I can't tell you. I don't have those results to show you or share because they need to be published first. But this is a preliminary experiment, and what it showed is that just by adding a little bit of specifically, the specific color spectrum of light to the inner canopy of these cannabis plants, enhanced, in all cases, enhanced the expression of terpenes and cannabinoids, the compounds of interest, at least at a cursory glance. There's obviously a lot more to the profile of medical compounds of interest in this plant, and our studies since this was published a couple of years ago, our studies have sought to look more deeply into the profile of compounds and come away with a clearer appreciation for Meshulam's entourage effect. We don't really understand that yet, and there's an awful lot to do. So we can do an end run on that. We don't absolutely have to know. We can provide, with high fidelity environment, we can provide a standardized commodity, a medicine, that I'm confident will reduce the error bars on the clinical trials thereafter. Next slide, please. So this is the end. We've just borrowing the space exploration challenge, borrowing from the technologies pushed by that challenge. We've come to a point where now we can quite routinely standardize the profile of especially the compounds of interest in cannabis and a wide range of other plants. We're looking at a couple of cancer drugs and the food crops that keep us fed when we're traveling to Mars and beyond. So I'll leave it at that, Keith, and I know you probably got
always a question fascinating. And now, thanks so much, Mike. There were some questions that came in, and actually you along the way were answering a number of them, but I wanted to touch briefly just to wrap it on 2 that I think are actually connected. One was about broad comment about the effects of microgravity or gravity on plants and the in some of your experiments, and air pressure in that sort of environment. And I'm remembering, I'm having a moment where I think you showed me spinach that you growed under Mars vacuum. So that's the kind of question. Any comments about that?
Yeah, gravity, plants have the capability to replace up with where the light is and down with where the water is. So we've grown plants on the International Space Station and all the way back to the Russians on Mir. It's something we can do relatively casually now. All of the work that we do in space exploration life support at Guelph are aimed at planetary bases like the Moon or Mars. So there is an up and a down there, happily, and I don't have to deal with microgravity and the technology pain in the ass that results in. So that's a good, and in any case, the requirement for the mass and energy cost of a biological life support system. So growing food on a spaceship, for example, in low Earth orbit, or on a transit mission to Mars, which is 6 or 7 months, we can carry enough bacon and Kraft Dinner to deal with that. Storage is okay. We don't have to have a huge garden of plants for life support under those conditions. It's just when we, especially on Mars, we get to Mars and we have 5-10 year exploration missions looking for life, which, by the way, we're going to find. You heard it here first. So those conditions will require that we mitigate the resupply requirements from Earth. And that means you have to start making your own food. And that determines, you all saw The Martian. That arithmetic that Matt Damon was doing is exactly what we work on in our program. It determines, how far you can go and how long you can stay. So the gravity issue, which started this, it is not an issue for us. And it will never be a major issue in biological life support, because you don't need it in low Earth orbit. You're only, a few hundred miles away. Mm-hmm. So we can resupply low Earth orbit. We can even resupply the moon if we must. It's only 3 days away. So
it's close. FedEx at some point would deliver. And what if a prediction about Mars, Mike, would you say when we are doing these missions and on Mars growing, will cannabis be among the growing plants, would you predict?
That is a secret mission of mine. At the moment, I'm stuck on barley for reasons that you know well. It is a precursor to single malt whiskey. Mm-hmm. But cannabis will come without any doubt in my mind whatsoever.
Well, thank you again, and have a safe rest of your vacation. Wish you well, and stay healthy, Mike. We need pioneers in the plant sciences like you.
Likewise. Thanks, Keith.